Yes, we are, Jean-Michel.
Awesome. So a very warm welcome to the first class ever of how to grow almost anything. I'm really super excited to have such a fantastic group of faculty joining us and such a wonderful long list of labs participating. So what we're going to do today is we're going to start with Neil Gershenfeld doing an introduction of Academy, Fab Academy, and how to grow almost anything, or Bio Academy. That will take not very long, but we'll go to the sort of overview. We'll then hand it over to David Kong, who will take us through the current classes and the current dates and the current faculty for all of you to be aware of what's going to come. We're then going to go to George Church, who is going to sort of give us a brief overview of the work he's been doing and the general content, the implications and the application, if you will. And we're very happy to be joined today by Megan Palmer, who is going to present us her apartment. She actually is going to talk about ethics and biosafety in conjunction with George. And I'll come back to you with David Kong, and we're going to go over any questions you may have as a site or as a joining lab. Hopefully we can answer them. If we can't, we're going to note them down and we'll try to answer them as soon as possible.
Jean-Michel, you dropped out at the end.
I said at the end, I'm going to come back, me and David Kong, and we'll answer any questions labs might have, or at least try to answer. And that's all for me as introduction. I'd say take it away.
Okay, welcome. I'm Neil Gershenfeld. I direct CBA at MIT, which grew into Fab Labs, which grew into Fab Academy. I'm delighted to see here, I'll do the count. Right now we have 29 sites connected, so we really are at capacity of everybody participating to the bridge. It's fully symmetrical. Everybody sees and hears everybody. So let me encourage you to join. This isn't a lecture. This is very interactive. I'm going to take 15 minutes to relate digital fabrication, Fab Labs, Fab Academy, and how to grow, to understand where this comes from and the arc around it. And to frame it, this is an event, I'll add a link to this back to what I'm going to present. I did with a very interesting president of ASU, Michael Crow, on the question of our universities obsolete. And the answer, my answer was 50%, and the 50% is exactly what we're doing today. And so this is a longer program with me and Michael talking about the educational implications. So I want to start by explaining how digital fabrication relates to this class. Claude Shannon wrote the best master's thesis ever, where he introduced digitization, and the heart of what he showed, if you haven't read his thesis at MIT, I really recommend it. What he went on to show is, if I send you a symbol to communicate as we're doing today instead of a wave, for a linear increase in the physical resources representing the symbol, there's an exponential reduction in your error in decoding it. That's called a threshold theorem, as long as the error is below a threshold. That exponential scaling in error rate with physical resource is the heart, is the deep meaning of digital. That's why we use digital. A larger group, including John von Neumann, applied that to computing, showing you can compute reliably with an unreliable computing device by computing with symbols. MIT made the first NC mill in 1952, but that's analog. There's no information in the material. The real model for digital fabrication is the ribosome. Chemical synthesis has a yield of, say, a part per 100. Elongation in the ribosome has an error rate of maybe 10 to the minus 4 because of the internal state. And DNA replication adds an extra error correction step and has an error rate of 10 to the 8. That exponential scaling is because there's information in the system. You can detect and correct errors. So the ribosome, 4 billion years old, is the invention of digital fabrication in the really fundamental sense, not 3D printing or NC machining. And so 2 years ago, CBA ran an event with the White House on the science of digital fabrication, on this transition to putting information into materials. And then a year ago, with a number of people in this class, we ran a meeting on the relationship between that and biology, turning data into bio and bio into data. And coming out of those is a sequence of, first step in 1952 was a computer controlling a machine. The second step today is using a machine to make machine. And so these are all examples of using rapid prototyping machines to make rapid prototyping machines. So the machines are as flexible as the class with them. And one of the new classes we're going to be adding to this sequence is a class on machine building, how to make these sort of machines. And I'll come back to that at the end. The second step in is digitizing fabrication by coding construction with materials. And so this is nano, meso, and micro Lego. This was, let's see, I think I have an example here of, this is one of my students, Will, who's making like a mechanical ribosome. It's a mechanical microassembler that puts together bricks of microelectronic material to build functional systems with the ability to detect and correct errors in their construction based on digitizing fabrication. And then finally, after putting codes in materials, is programming the materials. And so this is the math of inverse protein folding, where instead of trying to figure out what shape a protein folds into, you ask how to design a code to fold into a desired shape. And in a number of different material systems, we're implementing that kind of coded folding. And so that research on digital fabrication is following a history of, in computing, there are mainframe computers, mini computers, high-disk computers, personal computers. Now we're going from mainframes of fabrication like the 1952 NC mill to fab labs, which I'll talk about, to the machines that make machines, and the research up to the Star Trek replicator. What's important in this history is in the PDP era, where the lab filled the room rather than a desktop, that was the moment when email, the internet, all of that emerged. And for digital fabrication, that's the point we're at today. So at MIT, I started teaching a class called How to Make Almost Anything. And that was driven by, I had all these machines for research, and it would take a lifetime of classes to learn to use them. And so this was aimed at just a few research students, and every year hundreds try to take it. And it's just shamelessly teaching all of these skills, how to use all of these tools to make integrated functional systems. And the students do amazing things. Kelly, the first year, made a device to save up screens and plays it back later. And Meejin made a dress that defends your personal space. And they do these wonderful, expressive things, really doing personal fabrication, very analogous to the introduction of personal computing. And so that class was so popular, the National Science Foundation asked us to do outreach and talk about what we're doing. And so in scales, CBA runs a lab with, say, 10 $1 million machines for research. Within that, there's a workshop with, say, 10 $100,000 machines to make machines. And then within that, there's a lab with about 10 $10,000 machines, which is what we use to teach the classes. And then with that, you can make $1,000 machines. And so as an outreach project, what we put together was this. This was a fab lab. This one is in Vestmannaeyjar off the coast of Iceland. And so this is about a $100,000 investment, about 2 tons of stuff. And it includes design tools, 3D scanning, 3D printing, precision machining for things like circuit boards or tooling for production, computer controlling with a knife, with a laser, large format machining for things like furniture and composite tooling, surface mount rework, embedded programming, the 100K version of the tools. And with all of that, you can make all of this. So boats, bicycles, furniture, healthcare sensors, computer terminals, robotic chessboard, boombox, production tooling. All of these were made not in a factory or not at MIT, but using the tools in these community fab labs at this community scale. And so then the unexpected thing is they went viral. And so from the first lab, they've been doubling every year and a half. We're up to about 500 now with the doubling time of a year and a half. We didn't plan that, but this growth is sites opting in to join the network. So in turn, as the network grew, we had the same sort of problem I had at MIT, which is bright kids of all ages would show up at the lab, learn skills way ahead of local educational opportunity, and then fall off a cliff. There wasn't a next step for them. And so we started the Fab Academy. Initially, it was just Fab Lab sitting in on the How to Make class at MIT, but then it got much too big for that, and we spun it off. And so the Fab Academy has now run for many cycles. The last round through, we had about 50 labs participating in it, and a few hundred students joined. And to understand the Fab Academy, it's a very different model. What we're doing today is not obvious. In computing terms, you can think of MIT as a mainframe. You come to MIT and get processed. And it's very effective at that, but it processes a few thousand people at a time. I really don't like MOOCs. Open classes MOOCs list as a testament of how great they're doing that they have a class with 1 million people in it. And I don't consider a class with 1 million people education in the way I understand it. It has a role, but in computing terms, it's very much like time sharing. There's a central educational mainframe, and you're a terminal. You're by yourself looking at a screen connected to the mainframe. And it has a role. It's useful, just like mainframes and terminals are useful. But it's much closer to BitNet's model. BitNet grew slowly because it was a very high threshold to add things to it. The Fab Academy works much more like the internet. So what I'm showing on the right is a work group of students. This one is in Barcelona, which, as I see, is connected today. And then there's this video bridge we're all using. So students have peers in work groups with mentors as a real working work group, surrounded by tools locally. Then we link globally by video and content sharing. But the video and content sharing is links in a global network of local labs. And so this functions much more like the internet than the BitNet. And so fundamentally, it's distributed, not distance education. And crucial distinction between learning at a distance from a central site and learning in a distributed network in the way we're doing. It's curated, it's mentored, it propagates global best practices, but it's distributed. And we found a great virtuous circle. The Fab Academy each year builds a cohort of collaborating instructors and students. It drives best practices through the network, and then in turn it trains gurus that help create new labs and help expand. And so there's a wonderful virtuous circle that's grown up around the Fab Academy. So that's been working really well. To then lead into today, there's a very strong overlap between digital fabrication and biofabrication and biohacking. In the deep sense, work like George Church or William Shih or Peng Yin does really is the frontier of digital fabrication, turning data into things. And so for the fundamental research, there's this profound connection, but there's also a very strong technological connection. A good simple example is any biolab is full of what's in the upper left of somebody pipetting. If you're well funded, like say George's lab, you can afford what's in the upper right, which is a $100,000 liquid handling robot that does what the undergraduate does. For $100,000 though, you could instead buy a Fab Lab. And so lower left is my student Nadia, who's one of the leads on the Machines That Make Machines, did a collaboration making a liquid handling robot based on the machine building we're doing. And that's a few $100s in parts. It doesn't match the specs of the $100,000 one, but it's a liquid handling robot. And so you can buy a Fab Lab, and then amortize it in the first liquid handling robot that you make. And there's many other examples of that, of making thermal cyclers and the range of machines, microscopes in a biolab with a Fab Lab. And so there's this great connection between, rather than buying a biolab, you can use a Fab Lab to make a biolab and then do bio. And so there's both the intellectual connection and the technological connection. And so given that deep overlap, it then led to the idea, starting about a year ago, to introduce how to grow almost anything. In the first How to Make class, I had maybe 5 roles of plumbing and exercises and lectures and research. How to Grow now, those are forked off into George Church overall leading the class. We have a dream team of really the world's best biologists covering a range of topics with weekly units. We have this great network of labs joining. We have a program team, including David and Jean-Michel, running it to extend How to Make to How to Grow as the next big step in Academy, the Academy of Almost Anything. The name was a joke, but really increasingly as a joke we're taking seriously. Now comes How to Grow as we begin to add classes to it. My 50% is the question of what do you need to be. So if I look out this way, this is MIT's campus, and only a few thousand people fit here, and space is very expensive. There's a very high overhead. And so what do you need to be here for versus what can you do not by yourself but in this distributed way in the network? And there's still some things that need to be at a place like this, but a lot of what you do at a place like this no longer has to be done at a place like this. And so for me, this class is really at the front line of to what extent can advanced research and education be done not by yourself but in distributed networks rather than in central sites. They don't replace each other, but there's sort of a natural nesting. In the Fab Academy, we've seen there's Fab Labs, then there's super Fab Labs that mentor them and help provide supply chain and regional support, and then there's these super labs that have much more advanced tools. And I expect it'll grow into that similar kind of nesting. And so this is both directly a class, but it's also really an experiment in, MIT, again, if you look out that way, assumes space, books, people, tools, everything is scarce, or Harvard. And so we have to pack into these little spaces. And changing technology really means you can distribute them. In turn, once a year all these labs meet. Last year it was in Barcelona. Barcelona has great design sense and 50% youth unemployment. The economy is just broken for a whole generation. They're doing this amazing project to put Fab Labs in every district in the city as part of urban infrastructure. So if you live in Barcelona, you can produce what you consume. At the last meeting, Barcelona's mayor committed to a 40-year countdown to urban self-sufficiency, which means Barcelona really, the bits come and go, but the atoms stay in the city. And so that expands from just digital fabrication to things like energy and food and the whole bio life cycle. And so there's a profound need for a range of biotechnologies to meet the Fab City goal of urban self-sufficiency. In turn, a few weeks ago, many of you were in Boston for this wonderful event, Fab 11, where we brought 78 countries in. We had these dream teams of roboticists and biologists and politicians talking about all of these connections, and many more cities joining this initiative for urban self-sufficiency. In turn, the next one in the series is in Shenzhen in 2016. When you go to Shenzhen, this is where they make and you can buy Samsungs or Samsungs or Samsungs, or both made there, often by the same people. This amazing engine of invention. They're transitioning from mass to personal fabrication. So they help build the things that help everybody else build what they build. And this is where the graduation for this How to Grow cycle will be. And so finally, there's background for what I covered in these pieces. So it took a couple cycles for the Fab Academy to settle down, for the content and the infrastructure. Now it's humming along really nicely. In the same sense, this first time through is going to be bootstrapping, building the content, building the workflows, trying the exercises, but it's very much following the scaling curve of Fab Academy that has since been growing exponentially. And then finally, one of the interesting things that happened in Fab Academy is I went to my colleagues at EDUCAUSE that runs .edu about the accreditation, and we worked with the accreditors, and they said, we love what you're doing, where are you located? And I said, yes, and they said no, meaning by law, accreditation is geographical. You accredit a physical place. There's no notion of accrediting a network. But EDUCAUSE said something really useful. They said we should pretend. We should make up a credential and give it teeth by students building skills-based portfolio that document their ability. So in the Fab Academy, exactly what we do is we give a made-up accreditation but it's backed up by skills-based portfolios of students documenting mastery. And what we found is nobody ever asks where the accreditation came from, because nobody knows how to accredit this. What they ask is, what do the students know how to do? And so for advanced education, for employment, for investment, what they all care about are these portfolios. So each of the students is going to be building portfolio of mastery of all this material that we're teaching. Okay, so this is both the class. It's a meta experiment in advancing research and education, and it's a technological step in working together on this recursion of fab labs making biolabs. So that's how to grow. Any questions or comments?
I may have missed it, Neil, but what's total enrollment in How to Grow?
Let's see, I'm going to answer in two ways. Right now we're at 22 ports connected, and I think, Jean-Michel, how many labs?
Well, we're currently at 26 labs joining globally.
This is Anna France, by the way, for those of you that don't know me.
So Anne has helped manage the whole life cycle of people going through Fab Academy. Yeah, How to Grow grew very quickly. We've almost been beating people off with a stick who want to participate. We don't want it to grow too fast in this first bootstrap cycle.
This is actually, I think, going to happen with this as well. There's already a lot of people who want to join as a student, so we'll have to decide how big a stick we get out next year. If there's no more questions, and we can take more later, I think we're going to go on, because also for time, George won't be able to stay very long.
Yep, so I'm going to drop off. I'm going to mute now, but let me both welcome everybody and say just how happy I am to see this next step happening. We had just furtive glancing speculative meetings with Dave Kong and George Church, and I'm delighted to see it operational, and I'm very happy Jean-Michel stepped in to really organize it to make it happen. So have fun and good luck to us all.
Thanks, Neil.
Thanks, Neil. David, for time, when does George need to leave? Should we go to George first and then to you, or is it okay?
We're okay. George's got time.
Okay.
So Jean-Michel, can you hear me okay? Can everybody hear me all right?
Yes, sir.
Okay, yep. Great. So I'm going to push this over to you guys so you can see my screen. Is the content, can you see the content?
We can see it.
Okay, great. So Neil, thank you so much for the fantastic introduction. My name is David Kong. I am a synthetic biologist at Lincoln Laboratory at MIT. I'm also a community organizer based out of Cambridge here. And I was also, I did my PhD at the Media Lab and as a part of the Center for Bits and Atoms. So it was really kind of interesting to see the whole development of how to make almost anything. And also really to see, in many ways, the birth of synthetic biology and this kind of new era of engineering biology really emerge in the early 2000s, partially at MIT. And so for me personally, it's a great thrill to be a part of How to Grow Almost Anything. And to Neil's point about distributed systems and really being able to think about how to get biology out of the traditional laboratory institutions, the corporations, and the academic institutions into spaces like yours is really a great thrill. And so I'm actually presenting here in Cambridge at EMW, which is my community space, and we've got a room full of students. If I had a webcam here, I'd flip it around, but we've got a packed house here. So thank you all for joining, and really, really excited to be a part of this. So again, the course is being led by George Church, who's sitting directly to my left. Jean-Michel has just been an incredible rock star in pulling together and organizing everything. And again, we have a really just fantastic lineup of speakers who are going to be teaching. And so I'm just going to spend the next few minutes going through each week of the course and what you'll be learning and the type of material you'll be exposed to. And then just concluding with a couple of brief logistical remarks before passing it over to George. So We're starting today. This is August 26th. Again, a really historic day for us to really kick off this first class. Neil, you've already heard from, but I'm personally really thrilled and honored that Megan Palmer from Stanford, formerly Berkeley, transitioning to Stanford, is able to join us. Megan will obviously be speaking quite a bit later. Hi, Megan. Megan is a world expert and a real leader and thinker in policy and practices in synthetic biology and thinking about how we can really bring these tools and develop these tools, but in a responsible and ethical way. And so that's going to be a really big focus of today's class, and George will talk a lot about this as well. As how can we, I think, Megan, I'm going to steal a little bit of your thunder, but I think I saw the title of your slide, which is how to growing good, right? How to grow good, which I love the title. So that's going to be the major theme for today, and hopefully from both George and Megan, we'll really get some foundational concepts and questions about this whole art form of being able to engineer living things, and thinking about the safety, thinking about the ethical concerns. So as we progress through the course, we can refer back to this kind of foundational class and making sure that we're asking ourselves the right questions as we develop and learn the different technologies. So next week, we're thrilled to have William Shi from the Wyss Institute join us. William is, again, a world expert and a pioneer in DNA nanostructures. So as Neil was describing, going from data to things, but specifically leveraging Watson-Crick base pairing, G's hydrogen bonding with C's, A's hydrogen bonding with T's, and leveraging that programmability to create complex three-dimensional structures. So this is kind of the real fundamentals of DNA basics, but we'll be learning some really cool techniques about how to actually make some very complex programmable structures of DNA. Class number 3, September 9th. We're extremely happy to have Kate Adamala, who is currently a postdoc in Ed Boyden's group at the Media Lab, but is joining the faculty at University of Minnesota. And she's going to talk to us about synthetic minimal cells. So again, how to grow almost anything is very much a construction course. We're learning about how to use biology to build and manufacture things. And so in this class, we're going to learn about how cells work, but we're going to learn about how to actually make synthetic cells. So I'm using the materials and guts of cells, but encapsulating them in materials like lipids to create self-replicating machines that don't exist naturally in nature, but are going to be our own creation. So Kate's going to lead us on that. And then in the following week, Joe Jacobson, who again was my advisor at the Media Lab and is a professor at MIT, he's going to lead us on a class on next generation synthesis technology. So again, DNA synthesis, the ability to go from data to synthetic DNA molecules, which again encode functions that when put into an organism can reprogram it to do something new and valuable. Really, DNA synthesis is that core driving technology, and Joe has been one of the pioneers and leaders in developing new advanced ways to synthesize massive amounts of synthetic DNA, and was also involved, along with George, in the founding of a company called Gen9, which today is one of the leading DNA synthesis companies. So extremely thrilled and happy to have Joe leading a course on next generation synthesis. Class number 5, we are going to shift into the theme of biological production. So we'll have learned how to manipulate DNA, how to synthesize molecules. And in this particular course, Patrick Boyle, who is part of the Ginkgo team, is going to lead us on a class on how to actually think about encoding DNA molecules that can reprogram organisms to create things, to manufacture things for humankind. So again, Ginkgo Bioworks is one of the most successful synthetic biology companies on the planet. They recently raised, I think, like a $45 million round, so it's really happy for them. I personally, it's a real thrill. The folks that are pictured in the screen, along with Tom Knight, were folks that I went to graduate school with, so it's really awesome to see all the success they've been having. And Ginkgo's tagline is, the organism is the product. So they sell engineered organisms that make cool things, and we're going to learn about how to make our own engineered organisms. All right, so this is my favorite slide of this entire deck. So George is going to join us on September 30th for a full course on which he's titled Darwin on Steroids, Biodesign, Diversity, and Selection. George, I had not realized until I did a Google search on Darwin just how precisely similar you actually look to Darwin. So George, we're going to have to recreate, George, that's exactly, we're going to have to recreate this and do some photoshopping, but, and we'll have to find you a proper bowler hat for the photo shoot, but again, this will be really exciting. So George and for the next couple of classes, we'll be leading a section of 3 different courses and providing some more foundational framing on how to design biology. And again, Darwin on steroids. I'm curious to hear what that's all about, George. Okay, and then George, along with John Glass, who again we're extremely privileged to have. So John is part of the J. Craig Venture Institute, which again is one of the institutes on the planet that is really leading the world in genome engineering technologies. And so George and John are going to show us how to modify the genomes of organisms to, again, create new functions. And we'll be looking at a lot of different technologies in this class. So one of them will be a technology some of you may have heard of called CRISPR, which is really one of the, it's turning out to be one of the real revolutionary technologies in engineering biology, which allows and enables the very precise modification, site-specific modification of genomes. And John, again, has been involved in many big projects, including synthesizing the first synthetic genome for a self-replicating organism, Mycoplasma genitalium. And so this is going to be a really, I mean, they're all amazing classes, but this one I'm really particularly looking forward to see George and John really lead us into the world of genome engineering and all of the latest work there. In week 8, October 14th, we are thrilled to have Kevin Doherty, who is at the Wyss Institute and works with George. And we'll have spent a lot of time focusing on reading DNA, how to build things with DNA. And in this particular course, we're going to look at how to read DNA. Oh, sorry, we've been learning about how to write DNA. In this class, we'll learn about how to read DNA. So we'll dive into some of the latest and greatest sequencing technologies. So this is, of course, George made the comment that, his hope, and I think this is mine as well, that, in the planet, we can get the 7 billion people out there to have everybody develop some, literacy or ability to read and write DNA. So this is the reading portion. On October 21st, we're going to shift and zoom out. So we've been focusing a lot on molecules, single cells, and we'll start looking actually at tissues and organs. And so here we're really thrilled to have Nina Tandon, who was formerly a student of Bob Langer's at MIT, and is now the founder and CEO of a company called EpiBone, which is focused on growing synthetic bones. And so we're going to learn about artificial organs and how to build your own artificial organs. So I'll be focusing a little bit more on the human gut, but we'll touch on a variety of technologies and techniques therein. And so class number 10, we are thrilled to have Fio Aminetto from Tufts. And Fio has done some incredible work in 3D bioprinting, particularly being able to work with silkworms and actually engineering silkworms to actually be able to program and pattern and produce different types of materials, primarily silk, but we'll learn about 3D bioprinting generally and the ability to engineer organisms to manufacture arbitrary shapes in a programmable way. And next, November 4th, Kevin Esvelt from the Wyss Institute will be joining us. And so again, we've been shifting from molecules to cells to organs and organisms. And Kevin's going to start zooming us out onto the level of ecosystems. So Kevin is one of the pioneers and leading researchers on an area of research called gene drives. And so again, in the genome engineering class, we'll have learned a lot about CRISPR. And what Kevin has figured out with a bunch of colleagues is ways to actually leverage CRISPR technology to drive a particular trait through a population of organisms, so that after many rounds of sexual reproduction, you can actually essentially, again, drive a computer-designed trait through a set of an entire ecosystem of organisms. And that has really profound implications. And this particular class as well, Kevin is going to focus and discuss quite a bit about the ethics and the implications of this work, which again, Megan and George are going to lead us on that discussion to start with today. In class number 12, November 11th, I will be teaching a course on engineering the human microbiome. So again, many of you may be familiar with this fact already, but you are actually more microbe than human in many ways. If you, 9 out of every 10 cells of quote-unquote you is a microbe of some kind. If you added up the mass of all of those cells, you'd end up with something like 3-pound organs. So many microbiologists refer to this as the forgotten organ. It's just like your brain almost in mass. And so in this class, we're going to learn about this amazing ecosystem of organisms that's a part of your body, zooming in specifically on the gut, which is really kind of the rainforest of the body and actually the rainforest of the whole biosphere. It's one of the densest sources of microorganisms on the planet. And we'll talk a little bit about how to engineer the microbiome, and we'll explore how to make some artificial guts as well. In class number 13, November 18th, we are thrilled to have Srivatsan Raman, who again worked with George at the Vies Institute, but now is at UW-Madison. And we're going to learn about different types of biomolecule sensors. So you'll have learned about all these different types of ways to leverage organisms to produce molecules and different types of useful compounds. And here we're going to learn about different types of biological methods to sense those types of compounds and molecules. And finally, in class number 14, this course in a way ties back to biolabs making, or fab labs making biolabs. I'll be teaching a class on tool chains, automation, and open hardware. And so throughout the entire class, you'll have learned by this point a large number of different types of biological protocols, gone through different types of tool chains and methodologies for producing molecules, engineers, organisms, and so forth. And so in this final class, we'll get actually to work on making hardware that will leverage your experience from having gone through these different protocols to start building some of your own hardware. And this, in many ways, is a great lead-in to 2016. And I'll talk a little bit at the very, very end about kind of the high-level goals. But I do want to say that one of the strategic decisions we made in this version of this first version of How to Grow Almost Anything is really focusing on the biology. And so you know, there's a whole infrastructure build-out component that's a part of this. But this class is the one that we're really going to zoom into fab labs making biolabs. And finally, class number 15, after you've gone through all this technical work, we'll zoom back out again, just like we're zooming out today, focusing on intellectual property, what does the creative ecosystem for synthetic biology look like, and we're also leaving this in a way as a placeholder class. For sure, as we go through the coming weeks, there's going to be different themes and concepts that we may want to revisit, so we may end up repopulating this final class with a little bit of new material. And then finally, on December 16th, will be a final presentation. So you all of the students, you will be the stars of this particular class. And just to say, you know, for many of you that have gone through How to Make Almost Anything, this will be a little bit different in that the focus of the final presentations will be largely a design presentation. So the idea is, having gone through this entire course, being exposed to this different material, thinking about 2016, what would you want to grow? Okay, now that you know how to grow almost anything, what do you want to grow? And provide us with as detailed designs and schematics and processes and tool chains, et cetera, for how you would grow that thing. And for those of you that have the infrastructure and the capability to grow as much of that, that final presentation as you can. So actually try to manufacture and make that thing and present that in the final class. And so just a closing logistical remark here. The two major goals of this first go-around of How to Grow Almost Anything are, firstly, to ensure that at each one of your sites, there will be at least one trained local guru, one expert that's gone through the entire class, such that in 2016, when the class is taught again, that local guru will be able to lead instruction at that particular site. Okay, so that's goal number one. Goal number two is to have the infrastructure of your lab set up and complete, such that in 2016, you'll be able to participate fully in the class. And so how we design the course, we set it up in a way such that every single faculty has been assigned to basically create 2 different homework assignments per week. There will be a design assignment, which does not require any lab infrastructure. It'll just be software or basically non-lab based, all digital. And that way, every single student can participate in that particular homework assignment. And then faculty have been instructed to create a lab assignment. And so the idea is, again, some of you, and we recognize completely that every single space is at different levels in terms of their infrastructure and build out. And so recognizing that point, we hope that as the semester goes on, as we get towards the end of the semester, the different sites will have built up their infrastructure such that they can fully participate in the lab courses. And in 2016, for the duration of the semester, being able to fully participate. So those are just 2, again, key higher level meta points. And the final slide here is, this is just a photograph that I took from Fab 11 from the How to Grow Almost Anything workshop. Again, this is kind of really a historic photograph, I think, of our first session. And again, you know, it's amazing that we already have, 26 sites involved, and next year, I'm sure we're going to see a really huge, significant explosion in the number of students participating. One of the great analogies and metaphors from Sherry Lassiter, who is the executive director of the Fab Foundation, she said in regards to the Fab Foundation that working on Fab Foundation is kind of like building an airplane while you're flying it. And that metaphor applies extremely succinctly to what we are doing with this class. We are very much building the curriculum, figuring everything out as we go on the fly. And so today and for the whole duration of the semester, we are hugely appreciative to everybody for your patience, your flexibility with us as we build this airplane as we're flying it, or as we grow this airplane while we're flying it. But again, I personally am hugely appreciative of having met many of you at Fab 11 for all of your collective enthusiasm and excitement for this class. It's going to be a wild ride. Patrick Boyle, who's one of our faculty at Ginkgo Bioworks, he says biology is messy, which it is, and this course will be as well. But it's going to be a thrilling and adventurous mess, I guess. So that is all I have. I guess for sake of time, I'm going to pass it on to George, and we can save any questions about course flow and logistics for the very, very end. So George is going to present, then Megan Palmer, and then Jean-Michel and I will take over for more logistics at the end.
I'm just, before you take over, one quick note, I'm getting emails of people who are trying to make the slides bigger. Depending on what system you're using, you can either use the far end camera control, so these are like it's the keypad or the arrows to cycle through the windows, so they'll be bigger. If you're using life-size cloud, you can actually have a slider in the middle and you can make the slides bigger. So send me an email if you still can't do it, but try those two things. And
let me just clarify, with the far end camera controls on the keypad, if you set the far end camera controls number 2 on the keypad, every time you push number 2, it gives you a different window arrangement. And every time you push number 6, it changes which is in which window. And so you can use that to cycle to make it full screen. In addition, when we're done, Zerk, who's recording, the videos will separately have one window with full screen desktop and one window with full screen video.
All right, George, you're on. The floor is yours.
Okay, it's great to be here. Been watching this revolution of how to make almost anything and Fab Lab since very early days with Neil. And so it's great to see we're now moving to biology. This first slide, and I hope this can be a discussion, not just me talking at you. So I'll do various things to try to engage you. First of all, can I see a show of hands? How many people can actually see the slide at full scale? A couple, a few. Well, keep working on it, because I think I've tried to make the slides fairly simple, but obviously at low resolution, you're going to be depending on.
What we're going to do, we'll make all the slides available online somewhere else later, and I'm going to try for all next classes to have the slides be online before the class so people can actually look at it in their browser.
So I can already tell that there's going to be some motion, some animated GIFs that are probably not going to move. So that's okay, not too many of them. This is my conflict of interest slide, so if people want to know more about all the groups and people that have helped bring the work that we're doing into the world, it's on my website and it's also on this slide. David has already gone through the syllabus, but I wanted to have it up here for a moment. I'll come back to it at the end to make sure we've kind of touched on many of these topics, but in much more depth. But really at the core for this first introductory conversation, I'd like to talk about the strengths of both how to grow and how to make almost anything, their relative strengths, and the gaps and opportunities there, because there are some things that we can't make and we can't grow, and the hope is that by merging these, we'll be able to fill some of those gaps. And that's a great opportunity for everybody in this conversation today. And then since safety itself has an innovation component, I want to illustrate some of the innovative things that are happening at the cutting edge. But the cutting edge here is not necessarily something that is hard to, really requires gigantic infrastructure necessarily. Much of the innovation occurs on a very low budget. So I really think I'm very excited about this group. So we'll put safety as an example of an innovative thing you can do in synthetic biology, rather than putting in what might seem the more intuitive approach of putting ordinary safety synthetic biology first. And there's this design, build, test loop that we'll come back to from time to time. You might wonder why we talk about reading at all. We should just be talking about writing. I think those of you who've done enough of this fab know how important it is to be able to read to analyze what you're building and to check it out. And I'll make connections there. So the design of the design, build, test, analyze loop here. I'm just going to give you a few web links here, and you can poke around, and this will come up in subsequent classes. But we now finally, after about 5 or 6 decades of struggle, have pretty decent design tools for proteins. Proteins are the hardest, I think, on this list in a certain sense to get going on, but that works now, and you'll see some examples of that. Botson Raman when he comes up, for example. William Shee in the next class will talk about DNA nanostructures. DNA is a little bit blunter than proteins, but much easier to program. So cadnano.org is an example of CAD software there, developed in collaboration with Autodesk. We have genome engineering tools called Millstone. We don't really have developmental neuro and ecosystem tools that are at the same level as the first three, but here's some efforts in that direction. Here's an example of a feedback loop of synthesis and reading at the protein design level. So these are proteins that have been designed so that they have a new amino acid, not the standard 20 that most of us have in our bodies or in microorganisms, but a new one in the center there, which has 2 phenyl rings, 2 benzene rings, and then around it is a cage of actual electron density measured by X-ray diffraction in a crystal. So this shows the design. We built the organism. We built the protein, designed the organism and the protein as separate, and then proved that what we had was the correct thing. So that's what that cage is, the data, and the purple is the model that fits it. I just want to introduce a very basic chemical and biological concept, but in the context of an exciting synthetic biology. So the basic concept is that almost everything you'll be dealing with is made up of polymers, of simple monomers. So down at the bottom are examples of amino acids. The blue is the amino and the red is the carboxyl by convention, and then R is a variety of different side chains. So you polymerize these in a very distinct order. It's not like random polymerization, as Neil mentioned. There are these ribosomes. But what's interesting is they can make ordered polymers like the alpha helix and the bDNA double helix, and these have a certain chirality, a handedness, which is due to the chirality of the monomers, which you can see here. A mirror image of it is not superimposable by rotation and translation. This is a very fundamental concept, and it may be more obvious to this group than it is to certain other groups in entering biology. But the point is, this chirality, there's a heavy bias. Almost all of the molecules in the cell are chiral and in a particular direction. And while if you just make things in a chemical lab without special effort, there will be a mixture of both hands. So here's showing how proteins interact with DNA, and you'll see multiple reasons why the proteins are typically the catalysts or the machines, and the DNA is either the repository of information or part of a regulatory pathway. Now, this illustrates, again, this dynamic between reading and writing. And I illustrated a kind of a quirky example here of reverse engineering. So interactive time, how many people here have ever reverse engineered anything? Mechanical, electronic, anything? Not necessarily well, but okay, you may have taken apart a clock when you were a kid and tried to build one like it. This is an example of integrated circuit reverse engineering, which is very challenging. This article talks about all the way down to figuring out the kind of chemical structure at the level of the individual gate. And you can use all these different reading methods for getting at structure, and you can also look at function. You can read out the function by hooking up probes and taking high speed glimpses of electronics. We have completely analogous things in biology. We're trying to reverse engineer things, I think, in biology which are far more complicated than the most complicated electronic circuit. But it's the same sort of thing, structure and function. And one big difference in biology is almost everything self-assembles. So you have the advantage in a biofab situation, you will get used to this over the next few weeks, is you have a few advantages and a few disadvantages. We're kind of late to the game relative to electronics, which has been around for many decades. But we can inherit from electronics amazing computer-aided design tools and so on. We have the advantage that the self-assembly that I mentioned, you still have to do design to get it into the right range. We have the two kinds of evolution: the evolution in the past, where we have really powerful parts, which we can harvest if we can understand them, and then the evolution in the lab, where we can accelerate the process through design. So that's reverse engineering. Here is an example of trying to map human brain into electronics. I would say partially successfully. It's certainly successful enough that it can win more money on Jeopardy, but it's using 200 kilowatts to simulate a 20-watt brain, and it doesn't do nearly as many interesting things as I would expect a person to do. And so I would argue that in a certain level, molecular computing, meaning brains, DNA polymerase, ribosomes, and so on, is ahead of electronics in terms of energy costs. So this is kilowatts, or operations per kilowatt or per joule, and it's advancing faster. So most of the electronics is on a Moore's law curve, while biology is, or at least the kinds of exponential improvements in biology that we'll be talking about in this class are going a little bit faster. This is both reading and writing DNA. This happens to be for reading DNA, kind of a classic case where we were going at Moore's law curve. So the green is biology and the blue is Moore's law electronics. And around 2005, it changed radically. So this is a reminder these curves are not laws of nature. They're simply trend lines that have cultural components, and you can deviate radically in either a positive or negative direction. But anyway, the biology reading and writing of biological systems is going very fast. And so instead of taking 6 decades to get from a $3 billion human genome to an affordable, less than $1,000 genome, took about 6 years rather than 6 decades. And you'll see similar examples in writing DNA. Here's an example from the lab that I worked in as a graduate student here in Cambridge. For $6,000, we bought 10 oligonucleotides, which were 10 long. So these monomers I showed in an earlier slide, you can string them together and make something 10 long. We paid $6,000 for it. Now we might spend $500 for 15 million base pairs rather than 10. So it's about 7 factors of 10 over a 30-year period. And it's still dropping. It keeps better. And Joe Jacobson will tell you more about that. If you think about these as the bits and atoms, the bits of DNA are essentially A, C, G, and T. You can encode in principle 2 bits in those 4 bases, or you can lump them together and say A and C we'll call that a 0. So you have a little redundancy, a few options. So DNA reading and writing is not that tolerant of having long runs of bases. So various compression algorithms or having options of it allows you to encode it in a way that is compatible with reading, writing, and amplification. So this was an example where kind of to reverse the flow of technology from electronics and data to biology, from biology back, where I encoded a book, made 70 billion copies of it, of which 20 million are on this piece of paper, which Stephen Colbert, some of you may know, tried to eat it. But the point is, we can fit about a petabyte in a milligram, which is just a speck of dust. The entire internet can be fit into a small space. This has a long history going back to Joe Davis and his colleagues in 1986 encoded this piece of art, this 5x7 digital art, into DNA and read it by the DNA sequencing of the day back in '86, which was gel-based. And so you can see that this can be quite competitive eventually, not today. It's kind of a stunt today, where in red we have examples of DNA encoding in living organisms or in DNA in vitro, where this science paper in 2012 was a very large number of bits for the DNA. But we are now working with Technicolor and a band, Sri Vasari is working with a band called OK Go to put video and audio. This is for data storage of archival. This is not like a flash drive. Now, the revolution behind this, and Joe will tell you more about this, there's about 4 different ways to print DNA. Inkjet is one. One can essentially have something the equivalent of photomask, but dynamic. It could be digital micromirror array for photo deprotection. And this is typically done in a flow cell, and it's done under conditions that are kept away from ordinary atmosphere. No water, no air. It's usually done in a pure organic chemistry. And this has gotten us over 7 orders of magnitude, but I think we're about ready to switch that over to biochemistry, which is potentially orders of magnitude faster and cheaper. There are many ways that you can do error correction and assembly, and we'll talk about that in detail in the future. Again, this was a little bit of history. I worked on one of the first semi-synthetic plasmids in 1978. This is still present in many of the replicating components that you'll use in this course. Little bits of PBR322 are found in many modern plasmids where you can insert DNA and make it do what you want. It was originally used for making many of these proteins. You can engineer these molecular systems. I mean, the metaphor with machines is quite apt, but you need to be able to visualize and manipulate them. So this is from X-ray diffraction, as I showed earlier. You can make a model, and then you can manipulate this model. You can make design changes. In this case, we fused a DNA polymerase, which in code allows you to make copies of DNA. But in this case, we fused it with a pore, which is an unnatural fusion, but it allows us to read nucleic acids either by running them through the pore, where they can influence the conductance of the pore. This is sometimes called nanopore sequencing. It's one form of sequencing. It took many decades to arrive, but now it's one I think that you'll find very important in this course over time, because it really is enabling regular folks to do sequencing, and it should be something that you should be able to build this device as well. Fits in the palm of a hand. There's at least 2 companies that are in this space, Genia and Oxford Nanopore, but the Oxford Nanopore is the furthest along. It has incredibly long reads on the order of 500 kilobases. To put that in perspective, most popular sequencing right now is on the order of 200 base pairs rather than 500 kilobases, 1,000 base pairs. So anyway, that's just an example how synthetic biology can be used to help feed back on itself. It's a self-fueling cycle. Use synthetic biology to build tools that help you build synthetic biology and so on. This is some of the data from those nanopores. We don't need to go through it right now. You can run it in reverse too. You can not only go for a polymerase activity on DNA affects the pore activity. You can have a pore producing, modulating the ion flux, which then can affect the polymerase. So you can read and write to DNA or from DNA into biological systems as tiny as neurons, and you can make these little eavesdropping or recording devices, which I'm going to call ticker tape, to listen in on what the cells are saying. This is a nanostructure that is, we call a nanorobot because it has sensors, logic, and actuators. This was published in Science in 2012, and it opens up in response to the cell surface. William may talk about things like this. You can make sensors that open and close dynamically, and when they touch the right cell, they will sense that and open up the entire structure. So that's the logic is you have to sense on each side of the robot the same thing or two different molecules, and then if you do, then it does the actuation, which is deposits payload, which can be modulatory or killing, in this case, killing cancer. This is now scaled up enough that Pfizer is funding clinical trials on it. This is what our best guess is the minimum number of things you need for a cell. So if you are intimidated by the 4,000 proteins in E. coli or the 25,000 proteins plus other genes in humans, this would be the minimum set, and it's mostly focused on synthesizing proteins. So it's basically these proteins and RNAs make copies of themselves, and they're kind of obsessed with that process of going from DNA to RNA to protein. And so this is the ribosome itself. Most of the proteins are in here. It doesn't look very big, but it's very complicated with 54 different proteins. And most of the rest put amino acids, the 20 amino acids here, in a single letter code where F is phenylalanine. It's encoded by genetic code that we'll come back to again soon. Phenylalanine is encoded by things like UUU. So this is where nucleic acids turn into proteins, where the A, C, C's, and T's turn into the 20 amino acids in polymers. Little interaction. Show of hands. How many people are familiar with the genetic code? Anybody there? Yeah, quite a few. How many people can recite the genetic code without looking at a book or the internet? Okay, that's not going to be on the quiz. Not necessary. This course is not about memorizing stuff. It's about learning how to build things. It's conceptual. It's about concepts and where the cutting edge is. We mentioned CRISPR. This is an amazing discovery. It actually is kind of a validation of basic research where people found in 1987 in E. coli repetitive DNA. Nobody knew why, what it was. It was kind of boring repetitive DNA at first. Then later it was found that repetitive DNA had pieces of phages in the bacterial genome. People said, well, why would that be? Turned out it was a way of protecting the bacteria against the phages where they would remember what they had seen and survived barely. And then they would use that as a tool to then kill the phages that come in. So it was a way of, it was like an immune system where they literally would take a piece of the phage, store it away, and then use it later to cut it. Well, now fast forward from 1987 to today, and it's now a technology that you can use in almost any organism to cut the DNA wherever you want. So you can think of this as a sort of a search where you search through the entire genome, let's say human genome, 6 billion base pairs. And you don't do it in the way you might write a search algorithm in a computer where you start at the beginning and go to the end. You basically knock on each of the 6 billion doors in random order, sometimes coming back to the same one over and over, even though it's the wrong door. Eventually, when you find the right door, then you have an event, you cut. Or you can think of it as editing. You go randomly on every page looking for a match, and then finally you make a cut and change the text. We'll have a whole class on fluorescent in situ sequencing as a particular way of essentially looking at the molecules of cells, not just DNA, but RNA and proteins. Basically, this is a cell, a human fibroblast cell, actually from my body. You'll see more of that. And in the middle is the nucleus, and the outside is the cytoplasm. Every little dot there is a different molecule, and you can think of it as, you don't want to just look at the colors, you want to know the name of every molecule. And this is a way of the computer, the microscope can annotate the name of every molecule in the cell, protein, DNA, RNA, and in principle, small molecules. So that's really all we need to know about it for now, is that microscopy is no longer just looking at shapes. You can actually name everything, and that naming is microscopy. We have ways of making that very high resolution. You can ask multiple questions of each pixel, so you're not limited by resolution. Each pixel can have multiple molecules in it, but there's some very cool ways of improving your resolution. We have ways of sensing molecules, so that's a way of sensing that by imaging. We can also sense in ways that you can build into your biological systems sensors. So the camera you can think of as outside the system, but you can have molecules sensed within the system, and then the feedback loop can be very tight. And as I mentioned earlier, the biological systems are much more energy efficient and smaller. Essentially, everything we're talking about is atomically precise, which is not true of most electrical and mechanical engineering. And so you can have, it's much better to keep everything within the system if you possibly can. So rather than coming out of the system with a microscope and then go back into the system with some kind of synthetic chemistry at a macro scale, you keep everything as small as possible. So that's one strategy, and here's another strategy for sensing small molecules. And Boston Raman will talk about this a little bit when he comes up. Now, I've already shown one version of this genetic code slide before, where it was focused on the three-dimensional structure of the protein catalysts and the RNAs that make this up. This is a different way of looking at it, and it's actually a pretty important code. It's nearly universal, not quite universal, but every organism has this, from the lowliest bacterium to, the blue whale and forests and so on. And the only thing that I'm sure many of you know, that for RNA, the T becomes a U. So really it's A, C, G, and U for RNA, A, C, G, and T for DNA, and A pairs with U and C with G. Okay, I'm hoping that's all review. But in this complicated table, you basically have triplets. You have all possible combinations of these four bases, so 4 to the third power. You have 64 different triplets and 20 amino acids plus stop codons. So these are the 3 stop codons in the middle here. And you have things like UUU encodes phenylalanine. That was the first element of the code that was decoded, UUU for phenylalanine, or TTT if you're looking at DNA. And we put in boxes here 7 codons out of the 64, which we're in the process of changing by rewriting the code of the E. coli genome. So we'll be talking about this when John Glass and I talk about making genomes. And that brings us to this question of, oops, you have does yours take the same power that mine does? Bingo. Okay. So we've mentioned a little bit about CRISPR, this defense that's been used in bacteria, but it's also now used in all these different organisms and more. And it raises all kinds of interesting bioethics questions that people like Megan and I worry about. It can be used is being used to humanize pigs for transplantation, and one can make dozens of modifications now very simply using the CRISPR technology. So we're transitioning into a safety discussion. This is one way of accomplishing transportation. It shows the awesome power of automobiles. Part of what we're looking at, kind of at the other end of the spectrum of these highly engineered systems, for space exploration, and a whole new set of goals we have for being able to get off the planet, many of which have to be biological. To what extent can we build biospheres and have them populate the other planets? What do we want to take with us? What do we want to leave behind? So here's back to transportation on Earth. These cars, and these are all metaphors for safety in biological systems that we hope that you'll think about creatively and responsibly. I've mentioned modeling, but modeling can be used, modeling and computer-aided design and standards. You want isolation, protect the inside and the outside, and vice versa, protect the world from the cars from the world. It is not sufficient to implement safety ideas. You have to actually test them, and not just test them on models, but test them on the real cars or the real biological systems at scale if possible. You want to have redundant systems. Don't count on one safety. You can't, even though people say they're going to do the right thing, you still need to have surveillance because sometimes they accidentally or intentionally do what they shouldn't do. And so licensing is part of its education and making it clear what it is you're going to be doing surveillance on. And all of these have been proposed or almost all of them implemented in biological settings. These are some of the things that I've done in the setting of proposals that are now mostly implemented that deal with bioerror and bioterror, safety, security, and surveillance. And there's things that can be monitored, and hopefully that we will encourage the community to be as transparent as possible and really participate in this revolution in safety. Now, some, hopefully not in this course, but you'll be dealing with pragmatic safeguards of your own body, and hence people you may interact with after you leave the biolab. You won't be working there's these biological safety levels, 1 through 4. You will not be at BSL 4. There are only a few such facilities, and they work with things like Ebola and Lassa fever and things you don't want to be dealing with, recombinant or non-recombinant. But you might be working at BSL 1, which is generally quite safe, but it's not recommended that you eat or otherwise expose yourself to anything that isn't classified as food. We can engineer organisms. This can be a very enriching and exciting source of innovation. We can isolate organisms so that we have genetic and metabolic isolation. We can make an organism that's dependent upon a compound, a metabolite that's only available through organic chemistry. It's only available in the lab or in the industrial setting. So it really can't escape. And we do this by changing the whole genome, the genetic code. Again, we'll come back to this when we talk about genome engineering. We can put in non-severe amino acids. I already showed you an example of that. And you can make organisms that are resistant to all viruses. Principally, this is being done with E. coli, which is a major industrial microorganism, but in principle it can be done for any agricultural or even human cells. In terms of standards, I mentioned standards has a safety component. It also has, it makes it easy to exchange materials and know that you're talking about the same thing. And so as a consequence, the National Institute of Standard Technology and the Food and Drug Administration, which are responsible for safety in diagnostic and therapeutics, teamed up to create Genome in a Bottle. And this is kind of, I just mentioned this as an illustration of how standards interact with the sort of things you do, and also with a very open access and participatory community, very analogous to the FAB community. This is called personalgenomes.org. And when NIST and FDA decided to make these genomes, standard genomes, available to everyone in the world, so tens of thousands of identical vials of the same DNA or the same cells, so if you want to develop a new technology or a new medical platform, you could all be dealing with the same bottle. When they did this, they looked around the world for an appropriate cohort and and found that this cohort was the only one that was properly consented. So consent when you're dealing with human samples is a very big deal. And this was properly consented for re-identification, commercial use, and various other things. So this is right now the world's only open access data sets. It's also the only corresponding biobank for cells, including skin cells, stem cells, immune cells, and so on. And so if you go off and do human engineering, human cell engineering, it is very likely that you will use cells like these that have been properly consented. And these have the advantage that not only the cells consented, but there's genomic information about them, all kinds of omic information, neuro and imaging, and so on. You know a lot about the person behind these cells. Now, these cells can be turned into organoids. These human properly consented personal genome project cells can be turned into organoids, and representing almost all the major organs of the body in various degrees of abstraction or approximation. And this is some of the work from the Beats Institute and Don Ingber and Kit Parker and others. And you can see heart, lung, bone, kidney, liver. We're most excited about brain. Here's a cortical organoid, but they're also hippocampus and so on. Lung, where you have proper mechanical forces, multicellular context, air, blood, and so on. Pathogens, immune cells reacting to pathogens. All of these things are represented to variable degrees of significance and accuracy, which is improving each month. Now, this is a provocative list that I like to put up. People often think of human genetic variation that occurs in human populations as if it's rare, then it's deleterious. It's not most of the mutation thing, but here are some that have some interesting positive consequences. But it also illustrates there's probably nothing that is a complete net positive and no downside, and that's true. That applies to engineering, it applies to human genetics, it applies to synthetic biology in human cells. And you go down the list, you start looking at experiments that are done in mice that might be applicable to humans, where you have aging reversal, lower cancer, higher ability to solve problems, and do learning and memory. Very provocative list. The CRISPR and other means of gene therapies, CRISPR is not the only means, but gene therapy goes back at least to the early 1990s, and it was looking pretty good in 96. So what are we worrying about? What is it that we're obsessing about? And what we're worried about, what happened in between these two papers with the same first author, was that it initially looked like the 10 patients in the study of a severe combined immunodeficiency were doing really well. But after 3 years, 2 of the patients, called P4 and P5, developed cancer of the T cells. And this was due to fortuitous interaction of the LMO2 of the construct into near this particular oncogene called LMO2. A lot of the gene names you'll run across in bacteria and humans will be on the order of 4 characters long. So when you see 4 characters you don't recognize, it could be a gene name. Anyway, this particular oncogene was a very bad place for these things to happen. Happened quite rarely within a person, and not every person had it happen, but it happened in 2 people. And this caused a pause in gene therapy that is now more or less fixed. There are now 2,000 gene therapies in clinical trials, and many of them are emerging with full approval. For example, Vivera has been approved in Europe. That's therapy on humans in a medical setting. Those humans are running around free. They're not in cages. They're not in BSL-4 facilities. And another example of releasing engineered biology into the wild includes things like mosquitoes. Genetically engineered mosquitoes are being released by a company called Oxitec for trying to cure things like dengue fever and malaria and so forth. Right now they're using sterilizing the mosquitoes genetically. That isn't a full solution, and is that those eventually get overtaken by the fertile mosquitoes, and so you have to keep doing it over and over. It's a good business model, but it's not really a solution. Alternatively, there's this thing called gene drives that Kevin Estell will tell you more about, which allow you to exponentially spread through the population. It's not a virus. It's spread by mating a particular genetic element. But you can use it either to wipe out the recipient or to make the recipient species resistant to something else. So, for example, you can make mosquitoes resistant to malaria. You can also help agriculture by returning herbicide and pesticide sensitivity, and you can get rid of invasive species like rats, which are killing off a large number of endangered species on 700 islands around the world. More from Kevin. There are many other topics that are explored in this playful book, View of the Future and the Present. And I'm going to open it up for questions. So we're a little bit ahead of schedule now. On any of these topics that I talked about or anything that was on the syllabus that we didn't talk about.
George really is not very easy in responding to email, so if you have questions for him now, you should really ask him now.
That's not quite fair, but you should ask me now. Yes. I'm sorry I haven't made great contact with the people that are in the room, eye contact with people in the room, because I got this feeling that I'm out there in the world rather than here in the room. But we got a great crowd here. Yeah, there's somebody in the room. Say who you are and.
Hi, I'm Esther. I was wondering, so it seems like for a lot of the topics that we'll be covering in class, we are mostly going to be focusing on engineering bacteria, probably because they're simplest organisms to work with. I was wondering how much harder it would be for us to actually engineer plant cells, and whether there are like more legal restrictions on that, which is one reason it might be harder, or that we just don't know as much about.
So I'm going to ask the audience, should I repeat these questions? Show of hands, or yes? In the room. So it won't hurt to repeat them, if nothing else, to rephrase it and to make sure I followed it. So the question is, we're in this course, we're mainly going to be working with, what happened? We're working with with bacteria, E. coli. Because they're simpler. Would it be possible to work with plants, and what are the limitations there? I would say, first of all, that we're working with E. coli not just because it's simpler. It is an industrial microorganism. It's one of the main industrial microorganisms. It's not just a model system. So that's one reason. It is awesome. It has a generation time of 20 minutes. We have related organisms that have a generation time of 11 minutes, meaning it will double in 11 to 25 minutes. So plants are slower, so that's one thing. I don't think there's a gigantic containment issue. I mean, although you shouldn't be releasing any of these things into the wild, even if they're just an innocent piece of information. I think our ambition for this course in the future will be to engineer plants and humans. There are human cells. I have to catch myself on that, right? We don't want you engineering yourself just yet. So there are some safety considerations on human cells, for example. So a lot of these cells are derived from my body, and so I'm not immune to them. And so as probably not a good idea for me to be mouth pipetting at all, but certainly not my own cells that could have oncogenes in them and so forth. So also many of the cells you work with will have, that you might work with not in this course, but another will have Epstein-Barr virus in them as a way of transforming B cells. I want to be safe, careful about that. Yeah, that's some of the rationale that goes into why we picked the organism we picked for this first year. But it will, there's no real huge limitation. There's plenty of labs that can use other organisms.
And just as a quick note, it's okay, you can keep the camera on, I'll be a floating voice. But yeah, we will be doing a lot of E. coli work, but again, later on where with the 3D bioprinting, with some of the synthetic developmental biology work, we may be doing some mammalian cell work as well. So we are definitely going to play with other types of organisms other than E. coli. And again, a little bit of, some of this will depend on the infrastructure that you have in your space as we build out. So.
Other questions?
This is Anna France. Will there be any mention of fungus or mycelium in this? More plant questions, but.
Fungus, we will talk about fungi. I don't think there are any exercises. They're very important, both in conventional biotechnology. They include yeast, which is big in bread and beer, but they also include a much broader set of biomaterials and so forth that are subject to synthetic biology. And they have a replication time that among plants, animals, and fungi, sometimes it's called eukaryotes, they have the fastest replication time on the order of 90 minutes. It's slower than E. coli, but faster than just about any other eukaryote.
I'm really interested in, using it for materials, kind of growing materials and possibly machining them, experimenting that way. So if we address that, that would be excellent at any point.
We'll try to work it in. Almost all the organisms that we're talking about can be used to make materials, but you're right, the fungi already have a pretty good head start on that.
And just as a quick meta note here as well, again, because we're growing the airplane as we're flying it, if there are topics that become, that there's a larger growing consensus that we want to learn particular things or that we want to focus on something, we may be able to juggle the schedule a little bit. It may be possible to double team and introduce some new instructors, and definitely we can keep that feedback in mind for future years as well.
Just to piggyback on that, I'm sorry, go ahead.
Adding on to that, I think Fiorenzo, who will teach a class on 3D bioprinting, will actually also go into newly created materials and how to use them in processes like machining and making things with new materials. So there is part of the course that actually covers that.
Excellent. I'm really interested in artistic and building applications. Thanks.
George, I think you needed to leave at 10:30.
I'm a little flexible if there's.
Okay, then we can do more questions if there are.
We also want to get to Megan, which I think has some very relevant topics.
Okay, well, we can either do like we do in Fab Academy, which is take, as Neil calls it, 5-minute bio break, get a cup of coffee, and then go to Megan. Show of hands to do that? Yeah? Okay, so I'd say 5-minute bio break. It's now 30:35. We'll be back for Megan. And after that, we can take more questions and we'll go over class mechanics.
Actually,
thanks, George.
Where you guys get at? Can we take one? I'm going to take both for this sitting around the table.
You want to be in it too?
Yeah, I'm going to sit down. Take the hand. Awesome, thank you. George, thank you, man.
Power cable.
Oh, yeah, yeah, you can stick this. Let me,
I'll leave it in there so you,
okay.
or no, I leave
Make
it in
it-
there. Don't
I'd say that was a long enough bio break, Megan.
Can you hear me now?
I can.
Great.
You're not sharing any slides though. And now you are.
Okay, can you see the slides?
Yep, perfect.
Great. Just let me know, and I'm happy to start whenever.
The floor is yours, Megan.
Oh, great. Well, thanks for having me this morning. I'm coming to you from my apartment in San Francisco, from the living room. So if you see a cat wandering around, she'll be part of the presentation. I just wanted to thank. It's wonderful to be joining the first class, and I thank David and George and Neil and everyone for inviting me in. So as David said, I'm going to call this How to Grow Good. Generally was asked to talk about how do we think about safety, ethics within the context of what the Academy and How to Grow Almost Anything is doing. And so a lot about this topic, I just think generally about how we do good, how we understand good, and hopefully I can offer some provocations. But I am very happy to follow up at other times during the course when folks may have questions on how this comes to bear with some of the other things you're learning. Also, feel free to stop me if you have questions as well. So I wanted to start with a question, which is not a trivial one, which is how do we actually ensure that the development of biotechnology is overwhelmingly constructive? And by this I mean ethical, beneficial, safe, or at the very least not destructive. So how do we understand what actually being constructive with a new type of medium like biotechnology really means? And what are the actual practical processes we go about in delivering that? Some of you may have been following on some of the recent discussions about engineering of the flu. So this is a really interesting study in which we're trying to create some balance between understanding how, as we get better at engineering living systems, how we can both engineer them to be sort of within a parameter or meaning of control that we can respond to in a meaningful way. And so folks are really wondering right now, are we pushing the boundaries of engineering things like the flu in the case where we're not learning necessarily about how to respond, but actually creating things that we can't respond to. This gets even more difficult. Another news headline that's come up recently is around the ability to now grow things like painkillers, but also other opiates that might be used for both licit and illicit purposes in yeast. So there was a bunch of articles that just came out about homebrew heroin as an example where the balance of control, distribution, who can make what compounds over what scales is really being questioned, and it's really challenging our understanding of what it means to create a system when we can make useful products, but also control against their nefarious use. So I thought I'd start by just talking about how are we thinking about designing for good growth in biotechnology and in across these different labs. And I've been inspired in my work quite a bit by early days of growth of the internet as an example of sort of network distributed systems. I'm trying to think about the original people involved in some of the designs of these networks and trying to understand how did they go about understanding how their technical choices, their choices of protocols, their choices of the architecture, their choices of the individuals led to shaping of behaviors across these nodes, and really at a system scale beyond individual labs. And it's hard to know how much we can anticipate, the growth of these types of networks and systems. So this is the growth of the ARPANET in early days, and really very rapidly crossing huge geographical distances and causing a lot of questions about how we shape collective behaviors. As we know today, right, we're very interconnected, and it poses whole new types of challenges about how we interact with the technologies that we're designing. Biotechnology, I think, is the best example of needing to layer on an additional challenge on top of this, because we are dealing with a technology that's already taken over the world, and we're overlaying infrastructure and networks on top of globally distributed systems. And so how can we think about shaping our behaviors there? Let's see if I can do the interactive component as well, seeing you guys. Who reads XKCD? Hands? Yeah? Okay, many of you. So this is one of my favorite maps of trying to make a map of sort of online communities. Or as I think about it, it's sort of a map of the internet. And even though it's not accurate, conceptually it says something about, in these new types of domains that we're working in distributed technologies, who are the new types of organizations and entities that are shaping our behaviors and our norms. And these change and evolve over time, and so we're dealing with really non-traditional structures when we're dealing with these types of technologies. A map of a few years earlier might have had MySpace as a bigger territory, so this is changing over the time. And I think this is really one of the grand experiments that this class is working on is engineering really this global infrastructure for knowing how do we grow, how do we make things in a constructive way, how can we bake it into the architecture and the practices. And having that right in the first class means we're considering these things up front. So I think you guys are part of this, what I call engineering biology at social scale, really figuring out this as a design challenge, not just how do we design technologies to do good things, but mitigate against their potential risks and harms. A little bit of intro to who I am and why I'm working on these things. So I am a biological engineer by training. I was at MIT from 2004 to 2010, doing my PhD there on engineering of immune systems, but got very involved in the synthetic biology community as it was sort of growing up there. And after that period, transitioned over to working for the largest research consortium in synthetic biology in the U.S., SynBirk, that is where George is also an investigator as well, and was part of working with a number of people, including Drew Endy and others, to be responsible for, alongside the technology development, trying to come up with structures and strategies to deal with all the things beyond the bench. So anything from safety to security to intellectual property. And then also around this, we led a project called Community and Leadership, which was trying to ask how can we better organize our community to be able to ask these questions over time, and how can we really design the infrastructure to consider difficult questions around safety and security and intellectual property along the way. I now am switched from being a sort of engineer and scientist to being more of a policy wonk in a way, and I'm a researcher at the Center for International Security and Cooperation at Stanford University, and this brings together technologists and folks working in policy across all different types of fields to ask questions about what does it mean to use technology to make us more safe and secure and thrive. It's a really fascinating place to be. And part of my work is actually looking at the new types of organizations that are being able to shape what are standards and norms and safe practices in engineering. So I'll talk a little bit about iGEM, worked with them for a long time. I also work with national labs like the Joint Genomics Institute that are looking at developing large synthetic biology programs. and I started a fellowship program in responsible biotechnology leadership called LEAP that David Kong is a fellow in, and I think we'll have several of our other fellows, and I'm happy to talk about any of those later. So I thought I'd quickly touch upon sort of three general topics. Some of the first will be a sort of review of things you probably know, but it's a good thing to keep in mind, sort of technology trends, organization trends, and then how the combination of the technology advances in organization really come to bear on how we shape policies and sort of places where we're having big gaps in how we understand how to do things safely and ethically that I think are potential points that this class could really engage on and contribute to new thinking about. So I'm really excited to get your feedback on that. So one of the interesting challenges we all know is that as we think about biology as a technology, there's a number of different types of applications, all of it each have their own standards about what is considered safe, what is considered a good application, and it's really the diversity of different ways that biology might be used that is posing these challenges around how we have a behavior around one platform technology like biology as applied to many different domains. And I think the real shift that is causing stress in the way we deal with safety and ethics right now is something that's been alluded to quite a bit in this class and others, which is the ability now that we're technically advanced that we can very easily move between digital and physical domains when dealing with biological stuff means that it's causing us to need to reorganize the way we develop the technology, the way we think about the technology. And so this shift in now being able to actually rapidly move between digital and physical spaces and thinking about reorganizations of responsibilities and structures behind it is a really fundamental challenge that I think not many people have really looked at when it comes to this space, although it's been looked at in others. So one consequence of reorganizing the technology and getting better at technologies, we actually reorganize the way we work with it. So I spent about 6 years pipetting colorless liquids between tubes, and that's no longer what you need to do. But we're shipping things through to highly automated systems, in a way that can decouple the design from the fabrication. And it means that because we're organizing ourselves in different ways, the responsibilities around that are different. This is an example from the vaccine delivery, or sort of the vaccine group at Novartis, thinking about the future of vaccine development and deployment and mitigating of threats. And this is sort of a concept slide of how do we think about distributed systems being able to generate knowledge about new threats, centralized systems that can gather and curate that, and then deploy it in a number of different facilities all over the world. And really being able to understand how to do this in a safe and secure way is something that's just starting to be thought about, but is already being deployed. And an example of the key shifts in capabilities, this is again from that same group. What they showed is, for the very first time, they could collect the information about a new emerging flu strain, upload that, and develop a vaccine such that the rate-limiting step on being able to respond was no longer the actual technical development of the vaccine itself, but the regulatory approval of its safety. So all of a sudden, this sort of shift in whether or not it's really the technologies that are limiting our ability to actually deploy things in useful ways is shifting over to being much more of a social and organizational challenge. How can we develop systems that allow us to assess and monitor the safety of these systems in ways that keep pace with technical development. Another great example I alluded to before is key shifts and capabilities around production of a whole number of different types of molecules and new substrates. So this is another paper from some friends of mine, used to be in Christina Smolke's lab at Stanford, developing distributed biomanufacturing platforms for opiates. And all of a sudden you see really big shifts in who controls things like illicit compounds like morphine or elsewhere. Instead of it being grown in poppy fields in Afghanistan in illicit markets, it might be happening locally. And so there's a big assessment of whether or not this is going to be wholly constructive in terms of enabling more rapid, better tailored pharmaceuticals and therapies, or whether or not we're going to see the emergence of new types of groups working in this space. And I'd say that many of the claims made in this right now are really not backed up by a lot of knowledge. It's a lot of sort of gut reactions around what might occur. And so there's a lot of hard questions about assessing the possibility of different futures where we are able to easily engineer anything we want. And this is one that Kevin Esfelt will talk about later, which is the idea of now not just engineering end products using biology, but actually really being able to engineer whole ecosystems. And there's a lot of questions around what would it mean and how do we deal with these questions that are really of the commons around being able to shape ecosystems. So one of the things I'd love the class to think about and has a touch point, it really has less to do with how do you follow safety rules, but more around in the shifting places where new groups have capacities to engineer biology, where are we going to be shifting the responsibility for being able to respond to problems, detect problems, identify problems, and at the same time identify opportunities as we're shifting who is really controlling behaviors and norms and practices. So even within your own labs, as you develop these new capabilities technically, how do you also develop capabilities around your responsibility and awareness of other things going on in the system? Okay. I'm going to stop here, and hands up if you're following. We're good. Any questions? Good. Okay. So I'm going to shift a little bit talking about organization. So as many of you know, so this is the iGEM competition last year. We had one of the most inspiring places that I work. There's 288 teams, I think, across 36 countries. And so one of the things that I've faced in the last years is how do you help a group of biotechnologists that have never maybe worked in science and technology before understand collectively how to protect the sort of field, protect themselves, protect the environment, alongside developing new tools at the same time. And I think this is what each of the fab labs is going to have to come up with, is what are the systems that they can rely upon. This is the global distribution here of labs. Many different spaces are encountering the same challenge, so we're seeing not just with iGEM, but the DIY bio community really needing to figure out sort of with low resources, how are we going to make sure that we're doing things efficiently and safely and protecting ourselves over time. And they've been really great sources of innovation for new ways to think about safety, security, and ethics. Even places like this Glowing Plant Project crowdsource has been a place to think about new challenges for how we think about, this is a place where a large number of resources to do something have been concentrated, and they didn't necessarily have to ask permission. And so one of the ways that we've been working with Anthony and others is asking, how can we create good examples of good behavior that can then become sort of status quo when it comes to these new DIY projects. This is getting pretty serious as well with large companies like DARPA investing a lot, or large government associations developing large investments in biotechnology and the infrastructure around it, as well as a number of companies. So this is a number of companies working in this space. And a number of international attentions. This has obviously caught the eye of many governments, and many governments are trying to position themselves and asking, how can we direct this very complex, messy field with an expanding population of practitioners in a way that is most beneficial? And I think that is still a completely open challenge. I think last year there was something like 36 different governments that had national strategies around synthetic biology. So in this last section, I just want to touch base on the number of the types of strategies we've deployed in the past for trying to steer technology development in the life sciences towards good applications, and the types of oversight and processes. And part of this, I want you to think about what are the limitations, and what are the strengths of each of these different approaches. And in particular, life sciences and other technologies are often seen as having big dual-use conundrums, which means essentially as we get better at doing foundational things and manipulating these systems, they have both good and bad applications. And often controlling the use of them is more about the social systems around it than just the technologies. And biology poses a pretty interesting challenge because, again, there's naturally occurring diseases which we're also trying to work against, weapons and accidents, and these can often all be the same actual organism. So the same, in this case, anthrax, it's very, it's a naturally occurring disease which we have to protect against. It can be misused, and we can accidentally have problems with it. And there's really coupled safety and security challenges when it comes to understanding who is controlling these particular units over time. So key challenges here is in each of these strategies, how do we have our capacities for manipulating systems also keep pace with our capacity for detecting, analyzing, and responding? The gap between those is really sort of risk conceptually. This image actually comes from a 2003 synthetic biology report, even before major investments in the field were made. But there's a combination of both technical innovations and behavioral innovations that go side by side. Okay. I'm going to spend just a quick, I know this is text, but quickly on the word governance. This is a tricky word. I mostly avoid using the word governance when I talk with a lot of scientists and engineers that are working, but I actually think this is the perfect space to talk about it, which is if you think about governance as not just saying something that government does, but rather a place in which all sorts of actors are shaping the collective behavior of a field, then really what we're doing in terms of growing good is growing governance or designing for governance. So how do we design the technical systems to promote good behavior, good collective behavior over time? In the case of biotechnology, there's a number of different ways that we do this, right? We can think about controlling the development of the spread of materials. We can control personnel and their particular intention, and then we can control information. And again, because we're moving to a digital domain, this connection between distribution of personnel, decoupling of the materials and the information, and being able to spread that is forcing us to re-examine some of our strategies. This is a piece that I'm going to put up online. There was a really seminal paper that came out on a number of groups, the Craig Venter Institute and others, that looked at options for governance. So what are all the different types of actors, whether it's gene firms or fabs or government or otherwise, being able to take different types of actions to ensure biotechnology is being used appropriately, and also some different sort of design parameters. Is it enhancing biosecurity? Is it Is it helping safety? Is it protecting the environment? And also, is it making sure we aren't causing undue burdens to actually being able to creatively use the technology? And you'll see that these are really rough assessments, and I'll give you the full document online, but it's a really good example of trying to triage what is the set of things we can do to ensure the technology is being used in the best way. Here's some precedents and strategies that people are using that exist right now. And again, I can put some more resources online for you to look at these. Major one right now is the Bioweapons Convention. So this is, an international treaty enacted in the early 1970s, essentially with nearly all governments signing on to say we won't essentially develop weapons using biology. What's interesting about this is that there is no verification. So right now, it's basically governments getting together to talk about what's going on, and then they locally have laws to make sure that people aren't developing pathogenic agents or deploying them. And so there's a number of questions about how we actually keep these organizations in pace with their technical capacities. So this small office is 5 people in Geneva who are sort of keeping an eye out on the developments over time. One of the big ones that the life science community has relied upon are the development of scientific community norms. So some of you may know about the Asilomar meeting in the 1974, in which the founders of recombinant DNA, recombinant engineering, came together to really try and develop a framework around doing their work safely. And this is actually a Rolling Stone magazine article clip of the recount of the meeting. And there are some interesting tidbits in this. I can also post this online around what types of expertise is required to assess the risk. But this actually gave rise to the current structure used, the idea of different biosafety levels. So the development of these norms ended up getting encoded in guidelines developed by the NIH that have now been promoted by international groups like the World Health Organization for guidelines for how to work safely. Over time, we've also had to develop other guidelines around how to think about security. This is just going on right now, and really understanding, can we use the same types of groups that are assessing safety to also look at complicated questions around security and misuse. So in the U.S. is the national, yes?
Oh, sorry to interrupt, Megan. But if you just go back 2 slides to one more, forward to the NIH guidelines. Just one quick point here. Again, I think one of the really interesting aspects.
I can't hear you, David.
Oh, let me.
We can hear you in the class, though.
Can you hear me now? Oh, you can hear me in the class?
Yeah, I can hear you now.
Okay, great. Sorry. But again, I think one of the great parts about the class is, because of the international nature of the course, one interesting aspect of each of you and your spaces is kind of the different landscapes of regulations, and also kind of the different cultural takes on biology that each of your different sites have. And so I think Megan, in talking a little bit about what's happening in the U.S., it'd be really interesting somehow. And Megan, I'm not sure if you've got a homework assignment too, but it'd be really interesting to try and capture some of that information from all of you about the safety and the regulatory processes that you may have to go through to perform and work with biology as a technology wherever you are. So, because everybody's going to be different, so it'll just be that factor. I think will be a really interesting thing to poke at over the course of the semester.
Yeah, exactly. This is a great example where this is something that was first developed a long time ago, and now it's sort of being adopted and changed in different countries. And a big challenge is how do we share the information of what's working and what's not. And I think this class could be a great place to do that. And I do have some proposed homework. We can talk about it. And this security discussion is a good one. There's a number of countries that are actually, I would say, advanced from the U.S. in figuring out how to implement this, and we're still catching up. The other ways this has happened is we've had collections of industry groups come together to develop standards that are actually even greater than the ones that the government imposes. And an example of this is the International Gene Synthesis Consortium that come together to talk about how we screen sequences ordered from synthesis firms. And then a really big one that's often underappreciated is one of the biggest ways we're sort of controlling this right now, if you will, is actually import and export controls, not just of materials, but of information as well. And some of what is happening in this course and elsewhere, where we're really thinking no longer about shipping stuff across countries, but maybe sending information and then downloading it, it really sort of short circuits the systems in a lot of ways, and people are trying to figure out how do we modify export controls or shipping controls, or even whether or not we can want to classify, limit information. And so this is a big area where a lot of thinking is needed. And in the absence of that, saying these are all preventative strategies, one of the biggest things we need to do is bolster our detection and response capacities. There's a huge amount of money that is going into this, but also a lot of questions over whether or not even these themselves are posing risks, and whether or not it's sufficient given a lot of uncertainty over what might happen. Something to keep in mind is this: what happens when a problem arises, and something will happen. Definitely, this is one point of controversy was an experiment that went on 2 years ago of engineering avian flu and looking at the mutations that might cause transmissibility, and then publishing that information. And it really led to a lot of governments coming together, the scientific community coming together, and still going through a disagreement on whether or not these types of experiments are posing undue risks. And we need better ways to ask those questions. We also, as systems get more complex, there's a lot of problems around understanding whether old protocols are still a good idea, and how to make safety an ongoing challenge rather than just a burden. And a lot of the community responses lately that you may have heard about are actually in the absence of ways to go about thinking about this. Folks are just stopping research outright. This is a really blunt instrument, saying we're going to do a moratorium on all this type of research. No one's going to do it. International communities have come together, and once they do that, the sort of big question is how do you go and proceed? Next week will be the 1 year. A month from now will be the 1 year anniversary of, in response to that controversy, new regulations being put out by the U.S. government, at least, and we're seeing some of these in other places, essentially saying the government won't fund and support different types of research that they consider too risky. But again, we don't have the systems to assess what that might be in many cases. And we're really trying to figure out ways we can solicit expertise from new types of communities to be able to make these assessments. I'm going to just leave with, yep.
Sorry, just on that point. So, yeah, that was really interesting, right? So you're saying that the safety, in other words, the process for deciding what is safe, is that traditionally scientific communities that are actually the arbitrator? So is there kind of conflict of interest, like, oh yeah, what we're doing is safe, says we, and thus we can do it? Or, so is that
yeah,
what the
actually, one of the big challenges right now, I think, is that, so we have, in many instances, we have local oversight boards. So in this case, they're institutional biosafety committees that are composed of sort of peers at your university that look over grants and say this is good or not, and then they have to report up to their, the next level. And so far, the next level has been the National Institutes of Health, which is the largest funder of the field. So part of the debate going on right now is whether or not having that central oversight be within a funding body is actually, conflict of interest, and whether or not they have the teeth and the incentives, more importantly, to actually respond to problems when they arise. So I think it's a really great question.
Yeah, so, one interesting side note. So, we're here in Cambridge, and, the key regulatory kind of factor from a technological perspective here, and I think will be the case also internationally, you can do most of the work in this class without any special permit or anything. You can kind of just do it. It's kind of this gray, unregulated zone that isn't necessarily BL 1, 2, 3, or 4, but so for a lot of the work, you actually don't need anything special. But what we found is that, when the key technological moment is actually when you want to try to put DNA into an organism.
I'm still having you cut out, David, a little bit.
Try again. Are there folks able to hear?
I can hear you fine.
You can hear me fine? Okay.
Yeah.
So Megan, it may be a connection on your end, but anyways, what I was trying to say is, the key moment that the regulation comes in is actually when you need to put DNA into an organism, which is a critical step called either transformation or transfection, depending on what you're putting the DNA into. And in Cambridge, how they set it up, there's actually the same guy that was here in 1974 when Megan was talking about Asilomar and those first recombinant DNA meetings. In Cambridge, it's the same guy who's been here since 1974. So he actually would be a really interesting person to talk to, I think. And so you actually go through a permitting process here where local residents of Cambridge sit in a committee and you present to the public what you're going to do. So they actually introduced this valve that lets the public come in and say, like, hey, it's okay for you guys to do this community biology stuff or not. So again, I think one thing maybe we can do, Jean-Michel, as part of the class wikis too, is like learning about each different country's like set of regulations and what you guys all go through and everybody goes through collectively. I think that would be a really interesting data set to look at. So that's all. Thanks, Reggie.
What I'd like to do, and then I'll hand it back to you, Megan. I think part of the homework assignment for this week would be for everyone to concise description of the regulatory, of the rules and regulations in place locally in their country and maybe even in their city, as well as possible in the time we have, obviously, of those kind of systems. And maybe you can add onto that, Megan, at the end to sort of give a homework assignment after you've gone through your slides. So.
Yeah, and again, even
I'm
from
having
city
trouble
to
with
city,
the audio. Can you guys hear me?
We can hear you great. I can hear you. We can hear that again, yeah. But yeah, city to city even, like Boston to Cambridge, different rules. So that's an important distinction as well.
Okay. Still hear me?
Hear you great.
Okay. I'm having trouble with my audio, but I'm going to go in and just finish up this last little bit and then try and fix it. So the last thing I just wanted to talk about is I think a really great example of where we're trying to do some of the things that I think that this class could expand upon, which is in iGEM. And in iGEM, one of the things we realized is that this is a new type of organization that is able to also share different types of data of how people around the world are, what their rules are, and how they might culturally think about the types of benefits and risks of a new technology. So I've been part of iGEM since the beginning, and I guess on the board for safety and security, and also for policy and practices for the last several years, helping lead that. And one of the things that's interesting about iGEM is that it does enable this sort of distributed approach to looking at how you combine problems and people and sort of resource development and tool sharing also really integrated with the types of norms around each of those behaviors and incentives. So we've had to essentially evolve new approaches to understanding assessment of risk and surveying of risk over the course of as the competition went from one class to now 288 teams, and they have to now respond to some of the same questions I'm hoping you guys will think about, which is what does it mean to does the projects that they're working on comply with their existing institutional safety and security and ethics standards, which means identifying what are the rules, what are the norms, who's responsible, at your institution, in your city, in your country, and even in sort of international scales like the Bioweapons Convention. And the important bit of this is, secondly, identifying where there might be gaps or uncertainties or places where it's essentially the types of technologies weren't anticipated by the rules. And this is the most interesting bit because essentially it flips the problem of the people who are overseeing needing to come up with all the problems and actually ask the developers and say, are there things that you're developing that just, the rules that exist right now didn't even think they had to deal with? And then we've engineered in other systems to be able to oversee this and sort of do checks and balances over time. So we have an expert review committee that has people from all over the world who work on various local oversight boards and folks like the people at the Bioweapons Convention and folks who are working in health and elsewhere sort of review the questions that come in and the new problems and identify trends, and then also look at what teams would suggest. And then the other thing we do is we award prizes and create incentives for the best ideas and the best innovations that come out. And so there have been teams that have come up with better screening protocols or better ways to integrate design and safety, or even how to integrate the needs of various stakeholders into their projects. And then we also do some training, but really I think the best training is people showing good examples to each other, and that's what ends up happening. And the other thing we've done is really interesting, is actually have new types of partners work with us. So some of you who may know iGEM knows that a big sponsor of it is the FBI. So FBI, rather than trying to oversee or make sure people aren't doing anything wrong in this space, are actually saying that funding of these spaces to do their work well is the best approach to safety and security. And so it's a completely different approach. And so this is a great example. This is what I call know your local, friendly local WMD coordinator, or weapons of mass destruction coordinator. So essentially saying, get to know the people who, if something goes wrong, will have to respond. And it's developed a whole new type of relationship. And now we're actually seeing iGEM students go back to these really formal places like the Bioweapons Convention in DC and present their ideas to all the government experts because they're coming up with the new inventions. So that's all I had. Sort of technology, think about technologies for safety, organizations for safety, and innovating on both policy and sometimes in advance of policy practices that manage to combine the two in new types of distributed ways. And I have this homework, but I can talk about that later if you want, or whatever makes sense.
I'd love to hear the homework now.
Yeah, I think, Megan, what would actually be great if you could do a few things, put all the slides you've shown today on the page I created for the class. So that's the /doc behind bio.academy. So you can either send it to me, I could put it up, or you could do it. And I added just a header there saying class assignment. So best would be to sort of copy paste what's on your last slide there. But perhaps you could now take a moment to describe what you would like the students to do for next week.
Sure. So my understanding is that part of the assignment for next week is also to come up with the types of projects they might work on through the class, as well as something that's a little beyond beyond the class. Is that right?
Well, so that is sort of an artifact of us setting this up. I think that we can say we sort of, that might be an optional homework assignment that people can do if they feel like it. I think that what you've been talking about, I'm really very happy that you could make it today, is very important. And I really would like people to look into it at a very detailed level, because what we're doing here, it's very important that people realize what the applications and implications are. So I'd say your homework assignment has priority.
Okay. Well, I had this around the proposed project, so maybe I'll just describe it. But what I was hoping is that if you're thinking about the types of work that you might do through the course of the class and the course of the weeks, really asking a lot of the same, or responding to the same questions that we ask some of the iGEM students, and actually we do the same thing both in the national labs and in some of my other groups, which usually says, can you identify what the rules and the standards are that exist locally, nationally, and internationally? So in your region, what rules exist and who are the responsible parties? If you can, ask where there are uncertainties or ambiguities around these rules. So can you take one example of a given rule or standard around how to act safely that your types of projects or projects that you work on would cause some problems with? And so a perfect example of this is right now, a lot of our rules that we look at are things like, this organism is unsafe and this organism is safe. But that kind of breaks down when you're taking parts from one organism and putting it into another. So that's a really great example of a type of rule where doing organism-based assessment of risk might not work. And then if you were to come across that problem, do you know who the point person would be to resolve these problems? And this is a question because often we don't know. I know the folks in the U.S. who I would work with, but even the folks in the U.S. that I work with in the government and in the FBI and others often just don't know who the people are in other countries that are sort of taking the lead. And then I would say so you can do this in any way you want, really short form, written. And the bonus would be, are there principles of design that you would use to ensure that this community working in this class is doing something overwhelmingly constructive? So what types of principles of systems design would you use to make sure that we're doing things constructively? And maybe use this class as a case of, we have this problem with iGEM. I have thousands of students every year, and basically we need to figure out how do we ensure that they don't hurt themselves. So are there better ways that you can come up with that balance both allowing people to be creative, but also helping to protect themselves from the technologies?
Okay, great. So everyone, we're gonna put all this information up on the page for this class, and we will go over it in a few minutes, how I want each lab to set up a repository for documentation. But first, I think we can take a moment if there are questions for Megan.
Hello, this is Ferdy from Barcelona. Can you hear me?
Yes, I can.
My main problem right now is that I don't have a project yet, and I don't even know in what I can propose. Can you give us some hints in the direction of what our project might be?
I think you can also just choose an example project. So you can take one of the ones we've looked at, for instance, like can you make a drug or a chemical in a bacteria, right? Or you could say another one from the class, like could you grow bones? If you just can give an example, and work through the exercise there, that would be perfectly fine. But it would be great if you thought about something that you sort of aspire to do through the course, and use that. You know, you're not bound to it. It's just you know, try and think of something you'd want to do and where it might press up against.
So,
okay, thank you. Is there maybe a list of projects that we can use for getting inspired?
I might be able to do that.
I think fairly on top of that, I mean, it would be awesome, Megan, if you could make such a list. But on top of that, you could very simply look into what you need to set up a space that complies with biosafety level L1 and L2, for instance. Imagine you want to do experiments that are part of biosafety level L2. What can you do that? You know, what do you need to comply with to be able to do it, etc. So it could, like, setting up a space to participate in this class could be one of the projects you'd research on.
Okay, thank you.
That's probably a really good example.
Can you hear me? Jean-Michel, Megan? Yeah, you know, another thing too, again, the deck that I had, and I hope George shared his deck as well. You know, we described, and George's in particular, he went through a lot of different examples of really cool things that you can do with biology, right? So I think, you know, in thinking about what your project is, I think definitely knowing what the rules are for setting up a simple like BL1 lab would be a great base case. And maybe we can modify the homework text to specifically include that. So that'll be very practical homework because you'll need to do those steps if you want to get to a BL1 safety level. And then the second component of that could be, again, taking now that you've heard a really big swath of different types of things, you know, without having gone through the course, you can imagine to yourself, hey, you know, this is a cool kind of speculative thing that I might be interested in doing. I want to figure out how to grow my own organ, or I want to figure out how to you know, do X, Y, or Z based on the material that George and that we covered today. And then you can propose a project along those specific technical lines and see what the rules are there. So that's just a suggestion. And so Megan and Jean-Michel, by the end of all of this, maybe we can finalize the specific questions so that everybody will know what the specific ones are for the week.
Okay.
Cool. Megan, if it's okay with you, I'll disconnect your content channel and I'll push my desktop to show people where all this information is going to be.
Great.
Unless you have something.
And don't hesitate to reach out.
I'm sorry?
Oh, folks shouldn't hesitate to reach out for questions afterwards too.
Okay, great.
Yeah, that'll be, again, you know, kind of imagining the flow of this class. Again, please do reach out to the instructors when they teach. We've asked each faculty also, if possible, to get themselves a teaching assistant that can help them with the technical work. So throughout the week, if you have questions about the homework or questions about the content, please do email folks. We're here to help you and try to provide the answers to any of your questions the best we can. So just so everyone is aware of this, and probably everyone is, but just for redundancy, bio.academy.org/doc/classes brings you to the current list of classes. Each one of those will turn into a page filled with all the information we've spoken about. There will be class assignment posted on there. And so you'll be able to find everything we've talked about there shortly. I hope we can put up all the slides we saw by the end of this day in the U.S. And so this is where you will be able to find all the information. We've also begun filling the inventory page there. So this is a preliminary inventory, but you can start looking into that. And I think what we'll do is, coming out of your homework assignments, we'll use the information we collect, all of us, to put that into safety and regulations page, so people can go there if they want to know things specifically about these subjects.
Just to chime in there's also a shorter URL, how to grow almost anything, htgaa.org. little bit easier to find than the full URL, and it just redirects to the class page.
Other questions for Megan?
If there's no more questions, and again we can come back. I think we're gonna sort of end the class here, and then I'd like to take a moment to go over some mechanics, like documentation setup and sites currently joining. So Megan, it's really awesome you could join us today. Thank you so much.
Thank you. Great.
Oh, and then quick note. So Megan will be back for the beginning of next class. So next class, again, September 2nd. We're going to start at 9 a.m. And Megan will be there to review everybody's homework for the first hour from 9 a.m. till 10 a.m. Eastern Time here in the U.S. So thank you, Megan, again for coming today. And we look forward to interacting with you virtually over the week and then seeing you again a week from today.
Sounds good. Thanks, guys.
Thank you.
Awesome. So I'm gonna remind a couple of things to all of you, and then we're gonna end for today. Notably on documentation, most people already send me, no, actually it's not true. Quite some people are actually send me the information requested, not most. What I'd like each one of you to do is go to git.fabcloud.io/htgaa. I will again send this to you in an email later. You already have this in an email. There's a README there that explains you quite in detail how to set up a repository for your local lab. Please send me the link to that repository, and I will link it to the general repository. So there will be bio.academy.org/labs, which will have all the participating labs and links to the individual lab pages. So for instance, the one in Lima is already set up, so they simply have a page which is their index.html sitting in there, which is their home page. And this will be updated automatically. So the idea is that each lab, not each student, but each lab creates a repository, and you document your weekly work in that repository, push it yourself, and auto updates this whole system. The upside of this is that no one can break the entire system. Downside is that I need to link them individually, but that is just for the setup. So please take a moment today or tomorrow to set up your own repository for your lab and start documenting your work there. Next to that, just again to remind you, we have bio.academy.org/doc, which is for faculty. So all faculty will put the information there in their classes page, and there will be a growing list of inventory you can look into. So please bookmark that to be able to go back to it. Then a little note on finances. As many people, almost everyone should know this by now, but just again to remind you, we decided that there's $2,500 to be paid per lab this year, so not per student. Fab Foundation will be sending out invoices to each of you individually for that amount. Please take this very seriously. The money actually is very much needed to pay for faculty, to pay for organizations, to pay for servers. This is real costs that go into setting all of this up. So it's quite important to look into this. Again, inventory will be made visible, and we decided that graduation will be at Fab 12 in Shenzhen. So if you participate in the full scope of this class and you document well, we'll see all of you at the end next year in Shenzhen during Fab 12, and you'll receive the first diploma of how to grow almost anything in the world. So there's that. I think that's it for me. If there's any questions, or David, if you have something to add, please do so.
Yeah, so one quick note again, the inventory part, what's populated in that page right now is just really the core laboratory basics. So, you know, refrigeration, pipetters, some basic consumables, and so on. And again, you know, one of the key things that that's happening now, the faculty are still devising their homework assignments, so we're still figuring out the specific types of equipment that you're going to need for each class. What we're going to try our very, very best to do, and we're going to fail, but we're going to try our best, is to be 2 weeks ahead of time. So, for example, 2 weeks from today, we're going to have a class taught by Kate Adamala on minimal synthetic cells, right? So Kate, in the coming few days, is going to start posting materials that you'll need for the lab component of the course. And again, we understand that many of you may not be set up to participate in the lab component, but we're going to try to get Kate to potentially, since she's sort of our guinea pig, do 2 lab homework assignments, one really, really simple and basic and one a little bit more advanced. Like her advanced one is actually pretty intense. Like she wants people to make synthetic cells that express GFP, which would be challenging, I think, for like a graduate student in a fully equipped lab. So, but challenge is good too. So some of you may be able to even try to tackle that. But we understand that that it's going to be a process of building out the inventory. So again, we're going to try to stay 2 weeks in advance. So as you're working on the homework assignment for Megan and George, keep an eye out on that inventory list, and we can have Kate send out an email as well to say, hey, it's 2 weeks up, 2 weeks ahead. You guys should be ordering these consumables if you want to participate in the lab homework. Okay, so that's the flow we're going to try to keep. And again, we appreciate and thank you profusely for your flexibility and patience in advance as we go along.
I have a question. I know the inventory is rapidly evolving and changing, but is there any equipment that you know we will be using many times that we should look into right away, especially something expensive that we have to order?
So all of the materials that I put in the current inventory page, there's actually a really great article that actually somebody in this room, I think Linning, sent called "How to Set Up a Molecular Biology Lab for $1,000 from eBay." And so they actually go through kind of the core infrastructure, which I've shown here, which is refrigeration, temperature cycling, incubation for cells, centrifuge is a little optional, electrophoresis, so DNA separation, and then some basic consumables, and then pipetters. You're definitely going to need pipetters. So I would focus on the stuff that's on this page, and then also, you know, check out that particular link to that article, which I thought was really great as well. And so you can really get started for not too much in terms of upfront costs, just to do the very, very basics. And of course, you know, every single item on there, there's fancier versions of everything. And again, you know, it's possible to build your own versions of each of these tools, which we'll talk about at the very end of the class. But this is where to start, and as we update the inventory, we'll let you know. With specific things that you'll need for specific classes, we'll let you know.
On that, I'd like people, as much as possible, also to look in all the DIY versions of the things that are on there. And as our knowledge of that collectively grows, I'll be adding those to the inventory page. So ideally, there will be a inventory page at the end, just like the one for Fab Academy, which is a complete list of commercially available tools and products. And then there's the equivalent of do-it-yourself that makes your Fab Lab. Again, this is something I want to build with all of you. So ideally, we will have developed even some of these as a community by the end of this first year, and the next year we'll be able to use and build these tools ourselves.
Yes, actually, one great note, as you're building out your individual pages for your local sites, everybody should have an inventory page, actually. That would be really, really valuable. And then what we can do is use the class list. If people discover, like, hey, this particular model tool, I found a great version of this at a very low cost via this vendor, you can email that out to the class because everybody will be interested in that. And so that way we can all browse each other's inventories and see what collective equipment that we have. I think that'll be another really, a really nice community activity for all of us.
one more
Are there any
reminder.
other?
If
Sorry.
if you want to send out something to our participants this year, that's class 2015_HTGAA. Instructor_HTGAA goes to faculty, and the HTGA admin goes to me right now and the David for general questions. So please use those carefully. We all have enough mail, but that's what you can use.
One more question. I thought that there was an email that went around at one point about discount for, I believe it was OpenQPCR as a class. Is that something that's, still in effect, or something we should follow up on?
It's for the OpenTrons. It's for
OpenTrons, okay.
I'm pretty sure we can get it for the Kickstarter rate, which is $1,000, cheaper than the current rate. So I will ask them if this is still the case, which I'm pretty sure it is, and I'll send out an email with either the discount code or whatever they tell me to do, and I'll send that to the class alias.
Great, thanks. I mean, how important is that machine to have in the lab? I mean, obviously there's ways around these things, but
it's not very much.
Yeah.
Okay.
I would say it's very minimal, for this version of the class. But again, automation is a huge part of how we're going to transition from sort of the biological mainframe to the biological PC, whatever that looks like. And so, I think one of the exciting parts about Fab Labs making biolabs is, leveraging digital fabrication as a way to help improve automation. And so OpenTrons is a really great example of a first step towards open hardware applied to biology. But specifically applying that tool to the class, I think is not going to be quite as useful. Although later on, again, in the last class, I think if you want to build your own liquid handler, that would be a really cool homework assignment for the last class. So.
Okay, thanks, guys.
And I think it's similar as in Fab Academy. We try to create a set of tools and means without too much actually exactly indicating what needs to be done with them. So it might very well be that some people who take How to Grow Almost Anything this year or next year come up with super novel uses for those kinds of tools. So even if they're not necessary, do look into it and do share those links and information, when they become accessible. So if there's no more questions, I think we're gonna call it a day and say that for me, at least, this was a very successful first class of How to Grow Almost Anything. So
I agree, Jean-Michel. This is really wonderful. Thank you, and thank everybody.
Thank everyone for being present. See you next week.
Bye-bye.
Thanks, everybody. See you next week.